Good, thank you, Terry. So the picture, for those of you who have not been there, is the very large Texas Medical Center. So since the word cohort has been used about 7 bajillion times, I thought maybe we should pause and define what a cohort is. First, it's a sample of individuals, and it's a sample of individuals who share some characteristic. They've been identified through a place, or they've been identified through an experience such as the VA. I think it's also important to remember that it has some time frame to it. They were ascertained for time, and they're also followed up over time by definition. And the typical scenario is that they're measured early or monitored. In our case, we're quite enthusiastic about the use of mobile monitors, and then they're followed up over time for later outcomes. And that would be the typical cohort study. There's a lot of interest in whether a cohort needs to be representative. It does not. You don't need to have a representative cohort, or you don't need to have a cohort that's representative of the United States. We just need to have a cohort that we know from whence they came, so we can generalize the results to the population. So I think when we use the word cohort repeatedly, it's important to keep these definitions in mind. I've been very fortunate and honored to work in multiple NHLBI-funded cohorts and formed a consortium known as the CHARGE Consortium that has the goal to identify genes or genomic motifs influencing common chronic disease, particularly those related to cardiovascular disease, heart, lung, and blood diseases, and also aging. They're not all ancient, a word that was used earlier. Some of them are very newly established, such as the study of Latinos. The ERIC study is a large study, a multi-ethnic study or bi-ethnic study in that case, and the well-known Framingham Heart Study, just to name a few. The advantages of these individuals are measured for hundreds, if not thousands, of variables, and they're measured for these variables over time. They're not genetic studies. They're studies usually of the outcomes, and so they have both environmental and other exposures, and so it's an ideal platform to examine gene-environment interaction. The CHARGE Consortium now exceeds 100,000 individuals. There's about 75,000 individuals who are very, very deeply genotyped and or sequenced with very deep phenotypic information. All of these data, including genetic data and environmental data and phenotypic and outcomes data, are available via dbGaP. And I do want to thank the support of NHLBI and NIA for maintaining these cohorts as valuable resources for the scientific community. In the CHARGE Consortium, the working group, usually seated by young investigators, and I think that's a word we haven't used enough so far, is having these types of resources are great training opportunities for the next generation of biomedical researchers. Just to give you a taste of the kind of data, and by the way, on the bottom of the slide are dbGaP accession numbers for the data that I'll be talking about. So as of, I don't know about today, but as of contemporary, we have whole exome sequencing on approximately 11,500 individuals, whole genome sequencing on 5,700 individuals. The CHARGE Consortium has a very close collaboration with the Exome Sequencing Project, and when you combine the data, we exceed then 23 plus 11, 5,000 individuals. We have a very strong analytic infrastructure that I'll talk about in a little bit, so it really provides an outstanding analysis platform for investigational sciences. One of the examples of the work that this group has sought in collaboration with Richard Gibbs and Alana Morrison, whose pictures are on this slide, is really to try to tackle the analysis of whole genome sequence. Francis, when he introduced today, mentioned that an idea would be to have whole genome sequence data on all of these individuals. For many, it's an intractable data framework, and we're trying to make this seemingly intractable data framework accessible by providing annotation and analytical tools that would be available to the scientific community. This is one of two what I'll label as important slides. I think it's really important to not think about the U.S. research cohort as simply a collection, or we've used the word stitched together many times today, a consortium of cohorts. I really think we need to step back and ask how this collection of maybe existing cohort studies, bringing in clinical samples, and also as we identify needs, new sampling opportunities, how those can be much greater than the sum of the parts. As long as I think we have just heterogeneous groups of individuals that we're not paying attention to synergies, we'll fall short of our existing goals. So I think it's very important that we think not about a simple consortium of cohorts, but rather think about how we can create this community and have truly a national or U.S. Research cohort. This is the other, what I'm labeling important slide, is really there's enhanced opportunities if we can pull this off and make a true U.S. research cohort. The first is, we've talked many times today, that the U.S. research cohort needs to be participatory. They're not just research subjects, but rather they're fully integrated into the cohort or research enterprise. We're approaching that already today. The cohort studies have good community engagement and good outreach, but I think we can do better. I also think there's great enhanced phenotyping opportunities if we can bring together existing sample resources and create a U.S. Cohort, such as the use of mobile devices and also linking in real time these participants into the electronic medical record and the research platform. One of the things that hasn't received a lot of attention today, I'm a bit surprised, is this is also a great platform and infrastructure for clinical trials, very rapid and ongoing clinical trials. Since you'll have a deeply phenotyped group, you'll be able to very quickly ascertain potential trial participants. This may be heresy to say, maybe it's the second time I've used that word. It really is a platform for a national healthcare exchange or a national electronic medical record. So as people move around, we'll be able to move around and their electronic medical record will move with them. Also, the ultimate goal is to make enhanced community resources. I'm going to spend a little time, because it's something I'm very excited about, this virtual cycle between healthcare and research. As right now, most of us are in this phase of generating data and doing discovery, and that discovery phase, hopefully moves into translation. I think if we can generate and pull together this U.S. research cohort, we can basically build a cycle of which the translational information again feeds back into data in real time, and it creates more discovery opportunities that again creates more translational opportunities. That really, I think, is the promise of having this cohort in place for both the clinical and the research community. I'm going to give you a couple of examples. There's been a desire, what are we going to do with this thing? Here is a couple of very simple examples that we've annotated in all of the exomes in the charge consortium loss of function variants. And so we're able to look at either heterozygous or homozygous loss of function variants and then look how those phenotypes are related to common diseases. Here are the position of mutations in the HAL gene. This is blood histidine levels. You don't have to be a North Carolina statistician to see the people that have a mutation in the HAL gene have extremely high histidine levels. Histidine is a powerful antioxidant, and those individuals then are protected from small vessel disease. This model has been used in both research and in translation in collaboration with Helen Hobbs and Jonathan Cohen, PCSK9, something that's the poster child, in collaboration with Setkei Therasin, also in ApoC3 and NPC1L1, and then more recently in the product for LPPLA2. It also, I think it's important for us to realize that we need to gauge the individual labs. In the early days of the Genome Project, there was a lot of fear about how this very large mega project could absorb resources away from the individual labs. And what we see today is they really provide resources for these individual or smaller laboratories. So here's an example in collaboration with Jim Lupsky of really trying to identify oligogenic models and span rare Mendelian disease and common diseases. of having a case series of individuals with Charcot-Marie Tooth syndrome. We can identify in a subset the individuals that have point mutations, but then there's the remaining individuals who we don't have a molecular diagnosis. We then compare those individuals, in this case individuals in the CHARGE consortium, and you can see there's an accumulation of variation in the CMT genes. So by having a very large national cohort, it's an outstanding comparison opportunity for many, many other studies. So again, I think the two major messages that I'm trying to get across is, first, I think it's important to think about a unified national cohort or U.S. research cohort, not just simply stitched together of existing cohorts. And second, if we can pull this off, I think we're all going to benefit from this virtual cycle of really discovery, translation, and further data generation. So thank you very much.
Thank you. Okay, Tom and then Greg.
Tom and Ray?
Tom Insel from NIMH. At the beginning, Eric, you said that this was a way of getting at gene environment issues. So in CHARGE, what are the environmental measures and how do you address that?
So we have several gene environment working groups, alcohol consumption, dietary, exercise. Many of the studies have particulates, air particulates measured. And so we have many, many opportunities. That's just actually a small subset.
I loosed out NCI. One thing hasn't been talked about too much is what I would consider sort of the special sauce of a prospective cohort is the ability to get biological samples over time from the same individual. So you would be able to see disease, the phenotype before disease occurred. Especially important for circulating tumor DNA in the case of cancer, could be important in metabolomics, immune signals of incipient autoimmune disease. And we haven't mentioned that. I think it could dovetail nicely with the mobile monitoring, such that you get some signal, just like you would in a doctor's office, something's wrong, and that could be the trigger to go get a blood specimen.
I couldn't agree with you more. So all of these studies do have longitudinal collections, so we have longitudinal biomarkers that can be used both as outcomes and exposures, as you mentioned. I'm a big fan of using metabolomic data for both gene discovery and biomarkers. I also think it's going to be interesting, and I don't know what the VA is doing, if we're trying to link this with existing healthcare, what can we do as a group to think about using the doctor's office as a place where blood is drawn and how that could ever be moved into a biorepository. The logistics of that would be extremely complicated in the general practice setting.
Okay, we have one last clarifying comment, but just before we take that, if I could have Dave and Eric and Mike go ahead and sit up at the front table. We will have a panel discussion now. And Rod, go ahead.
Roderick Pettigrew, NIVIB. I wanted to respond to Tom Insel's question about environmental exposures and what could be assayed. Tom, virtually anything for which there exists a ligand, be it a chemical, bacteria, virus, particle, a toxicant, can be assayed. The platforms to do that, the technology could do that exists.